In Unit 7, we'll cover Chapter 9, which is Mendelian genetics. Before we embark, we'd like to recall some of the terms that we've been using. Recall the name chromosome. This basically means a single strand of DNA, but typically we call a chromosome this name when it's bundled up or condensed. On those chromosomes lie genes, and that means each segment of DNA along the length of that chromosome codes for a specific trait or gene. Remember that humans have 46 chromosomes, or 23 pairs. On those 23 pairs, the first 22 pairs have genes that code for, well, being human. In this slide, you can see some of the numbers on a few of the chromosomes for the number of genes they contain. Chromosome number 1 is our longest or largest, and it contains about 1,270 different genes. Now note that the 23 pairs come in 22 autosomals and 1 sex chromosomal pair. The genes that code for our traits are essentially the recipes for building proteins, and it's these proteins that go on to give us, well, our traits. They build structures as well as keep our bodies running. Remember all the variety of structures and activities that proteins are engaged in. Now each gene comes in its own particular form. Depending on what those are, we call each of them an allele. Each of us carry 2 alleles for each gene. We need 2 terms to be defined because we're going to use them a lot. The term genotype refers to what are the particular alleles that you possess. In essence, what is the genetic makeup you have that you're carrying in those genes. Your phenotype then is the physical manifestation. What do those genes make you look like? Are you tall, blonde hair, or what color are your eyes? Our knowledge of genetics is thanks to a monk by the name of Gregor Mendel. He in the 1860s spent decades patiently breeding pea plants in his monastery. It's thanks to him that we understand the passing of our genes to our offspring and what that means in a genetic sense, what we look like. Now, he happened upon peas and chose 7 very simple traits. It was lucky for him because each of these traits only had 2 alleles. One allele was a dominant form, so it would mask out any other alleles when it was present. And the recessive allele, well, it was recessive. It was sort of the weaker and only showed through when absolutely no dominants were present. Now each individual carries 2 alleles, and depending on your combination of dominant and recessives, well, that gives you a specific kind of name. The terms you'll need to know: homozygous. Homo means same and zygous means structure. These are the homologous type of structures that we talked about last week on the chromosomes, only now we're diving deep into the individual genes. So a homozygous dominant individual, or in this case a pea plant, has 2 copies or 2 of its alleles, both carrying the dominant form. In this slide, we're showing you the pea plants and the purple flower being the dominant. So if you're homozygous, you're carrying around 2 purple copies. If you're heterozygous, that means you're carrying around 1 dominant and 1 recessive. Remember, the recessive is hidden, so you'll still appear to be dominant or purple colored. Lastly is homozygous recessive. That's the recessive allele, and in this case, the white flower. White flowers only exist when 2 recessives are present. Now, when working with genetic problems, we assign letters to make it easier than having to write out the words purple and white. So, in this case, they've assigned the letter P. and the capitalized P is always the dominant allele. The lowercase p would be always the recessive allele. If you recall from last week, we could assign these gene alleles to the actual homologous pairs of chromosomes and separate them in meiosis. Well, Reginald Punnett, a mathematician and geneticist, who came up with an easier way than following through meiosis in every stage, he gave us a shorthand. He developed what is known as the Punnett square, and really it's just a way of easily placing the different alleles that exist on those homologous pairs of chromosomes in a box that allows you to predict what will the offspring look like if the parents donate each allele in their gametes. Now, Mendel's laws follow the same laws we learned last week in meiosis. The law of segregation, so we're going to split up the alleles into the individual gametes just like we did in meiosis. And let's not mutate anything, at least not yet. The law of independent assortment, if you remember, basically said when they line up in metaphase 1, it's random, so that we don't know which side of the metaphase plate the mother and father homologous pairs of chromosomes are going to line up. And therefore, when they do segregate, we're going to get sort of mixtures that will possibly turn out in the gametes. The independent assortment is talking about that the genes on different chromosomes are free to separate independently from one another. There is one violation to this law, and that's if you're specifically looking at two genes that lie on the exact same chromosome. Those are known as linked genes. For now, we're going to talk about genes that are independent from one another. When we're solving for monohybrid crosses, or crosses looking at one trait at a time, we need to first always start with the parents. Typically, you'll be given sort of like a story problem, and we need to identify in that story problem what the parent's genotype. Remember, that's going to be the actual genetic makeup, or in this instance, the actual letters that we're going to be assigning. In following with the purple plant, purple pea flower color, we'll identify this top first monohybrid cross as two parents who are heterozygous for the purple flowers. Notice if they're heterozygous, they have one dominant and one recessive allele. When we write their gametes on the Punnett square, we're going to simply separate the big P, the little p, the dominant and recessive alleles, in each box. Don't put two p's in a box when we're looking at the gametes. The gametes are put on the top row and first column, and one parent per side. So, say the father's alleles are across the top, and mom's are down the side. If those are gametes, recall you should be haploid, or only have one copy of each allele, therefore only one letter P. We then will go through and fill out that Punnett square by crossing or bringing together what would be those two gametes. And you'll notice you get parents that are heterozygous can get children that are homozygous dominant, that's the large P, the heterozygous, large P, little p, and we get two of those, and a recessive, little p. So when we do Mendelian genetic problems, always start with the parents, produce the gametes, create the Punnett square, cross those gametes, and then summarize the offspring. So in these two examples, when we ask you to summarize, think about what the two types of ways of summarizing would be. To summarize the genotypes, we would need to lead with the actual letters. You could say, in the first box, 1 big P to 2 big P to 1 little p. Those are genotypes. Phenotypes would be colors, or whatever the physical expression of those alleles are. So in that top example, that would be 3 purple to 1 white. Pedigrees in genetics are a visual representation of the family's genotype and or phenotype. So it's a family tree, but it's one in which we track a specific trait or set of traits through that family. Now this is a rather complex pedigree on this slide. We'll have you looking at simple ones in lab. For the complex one, the things just to start off with. Well, females are always circles and males are always the squares. And you color code depending on which trait you want to particularly follow. Now typically we'll be following things like diseases to see how they're passed down and move through a family. Therefore, we'll generally color code in such a way that we can see which individuals have the disorder or the disease and those who do not. Now in this particular pedigree, they're actually showing you even the heterozygous, and that means this one is showing you a little bit of the genotypes. The pedigrees we usually look at least in lab, will be more simple based on phenotypes. Diseases and disorders can come in both dominant and recessive forms. Some recessive forms that we see, or caused by recessive alleles, are things like albinism, cystic fibrosis, and sickle cell anemia. But dominant disorders do exist as well, like Huntington's, Alzheimer's, and high cholesterol. Now, one misnomer before I move on is that dominant traits are not always the most abundant. If that were true, well, we'd all have actually 6 digits instead of 5. Dihybrid crosses are when we look at 2 traits at the same time, and we put them together to see how the combinations together will look in the offspring. These are a little bit trickier. In this example, we're looking at the peas that Mendel grew, and in this case, we're looking for the shape and color of the resulting peas that each plant produces. So the parents in this example are completely heterozygous for both the shape and the color. So the big R represents the round, and the little r represents the wrinkled. The big Y is yellow, and little y is green. Together, each of those parents is carrying 2 copies of the shape allele, big R, little r, and 2 copies of the allele for the color, the big Y, little y. So both parents are completely heterozygous for both traits. That's not always the case, but let's follow this through. Now, the tricky part is finding the gametes. Before, when we were looking at one trait, we simply separated the big R and the little r and put them on the Punnett square. But now we need to sort of follow through what are all the possible ways in which the R's and the Y's could have ended up in the gametes due to meiosis. And to do that, the easiest way is to do something from math called the FOIL method. FOIL methods are basically just distribution methods. We're going to distribute each R in each Y in the combinations. So let's look at the word FOIL. The F stands for the first of the pairs. So that would be the first R and the first Y. In this case, that is the big R and the big Y. The O stands for the outer pairs. So the big R and the little y. The I stands for the inside pair, the little r and the big Y. And lastly, L stands for the last pair, little r, little y. Practice foiling lots of different combinations, because if you get the FOIL incorrect, then you'll put the incorrect gametes on the Punnett square, and you'll get the whole problem wrong. So do be very careful. Try foiling. The FOIL method allows us to get the gametes, and each of these gametes, as a check, should be haploid, have 1 copy of each allele for each trait, meaning 1 of the R's with 1 of the Y's. If you end up with a gamete with 2 R's or 2 Y's, retrace your steps. You didn't FOIL correctly. Place these gametes on the top and the side, 1 side per parent, and then begin to fill out the Punnett square boxes. You'll notice this is a 4x4, so we get 16 possible combinations that are different. So these are going to take you a little bit longer, but if you go slow, write very carefully, you'll be able to get it. You'll do several of these in lab. One quick note: if you don't like a letter, change it. These are only meant to represent the dominant and recessive alleles, the forms. If you can't write the 2 letters different, choose a letter that you can, because we really don't care about what letter you use as much as what it represents. So ultimately, what color will you be? What shape of piece will you have? So feel free to change those. Well, we kind of think Mendel got lucky. Maybe he planned it that way, that he chose 7 particular, very simple patterns of inheritance, meaning each of those 7 traits he followed in his P's, really only had 2 alleles, a simple dominant and a recessive. So that's a pattern of inheritance. Simple dominant recessive, though, doesn't account for all the variation and the different patterns that we see out there. We'll look at a couple of other patterns of inheritance. The first one is called incomplete dominance. And in incomplete, the actual dominant allele doesn't completely hide or mask the recessive. So the dominant homozygous individual, in this case, these are snapdragons, a red flower crossed with a recessive white flower will give offspring that are all heterozygous, big H, little h. Well, that's the same as Mendel's simple dominant recessive, but what's different is that heterozygous has its own unique phenotype. It looks different. It looks like a blended in-between trait. In snapdragons, that means the heterozygous are always pink. The second pattern we'll look at is called codominance. This means there's multiple dominant alleles. In the human blood example, the ABO system, 2 of our alleles are equally dominant. So when a person has both of those alleles, their blood is actually going to be a mixture of both of those. What are we talking about? Well, the ABO blood system are basically receptors on the cells of our red blood cells. When you have one kind of receptor present, you have the type A. When you have a different receptor present, you have type B. And when you have neither of those receptors, well, you're the recessive type O. You'll notice on the chart that the type A, B, AB, and O are really phenotypes. To actually write it genetically, we need to account for both alleles. In this chart, they're superscripting. Some people like to superscript, especially in the medical community, because that way it's very obvious that both A and B are equally dominant. Note that they are superscripted on capital I's. You could also just simply write those as big A's and big O's. Just note that the O is the recessive. So a person who is type A blood could be, for their genotype, AA or AO, hiding a recessive. The same goes with type B blood. They could be BB or BO. But the AB blood is showing that codominance. The only genotype that a person with type AB blood is AB. They're carrying both alleles. If you are the recessive O blood, that means you're completely recessive and you're carrying around 2 copies of O. Now, some of you might be asking about the positive negative. Hey, I'm A positive. The positive negative is just another way of writing a simple dominant recessive pattern. Pleiotropy is a condition in which one gene can affect multiple systems and structures. In the example here, you're seeing one gene, the gene for hemoglobin. That protein, recall, carries oxygen throughout our body. Well, if you get a recessive form of that hemoglobin, which is a mutated form, it means that your hemoglobin is misshapen, and that results in cells that are shaped incorrectly. Instead of being smooth and round, they turn into, well, sickle shapes, or little curved sort of comma shapes. These incorrect blood cells can break down, clump, clog, and even accumulate in incorrect places that have a large ranging effect throughout the body. Notice as you go down through the arrows, the huge number of systems that are affected. This is all because of one gene, and this is known as pleiotropy. Polygenic inheritance is really accounting for the great diversity in a lot of our traits. Think about skin color, hair color, height, weight, all of those things that have genetic components, but there is not just a black and white, just two options, there's multiple options. Well, that's because it's the additive effect of many genes, polygenic. Dominant recessive alleles do exist, but because there are so many genes involved, you get a huge array of different variations. If you look on this little graph, you'll note that the lightest color has all 6 alleles being incredibly light, but the darkest individual has basically the dominant form of the allele for all 6 genes. So we can sort of estimate how light or dark an individual will be by counting how many dominant and how many recessive alleles they are carrying. Now you could apply that to even your eye color. Think about the darkest of eyes that almost look black to the lightest of eyes which are almost gray, and all the variations in between of the hazels, greens, and browns. And don't forget the blues sitting near the recessive end. All these Mendelian genetics problems that we'll be having you do in lab are really to sort of predict and look at the variation that can be passed down to our offspring through sexual reproduction. And those gametes, remember, are the product of meiosis. But also remember from meiosis about crossing over. Crossing over gave us new combinations of genes on our chromosomes, right? Remember when we swapped the tips of some of those homologous pairs of chromosomes? Well, linked genes, if they're together on the same gene, always have to segregate together unless they're crossed over. So crossing over is a way for linked genes to become, well, unlinked in a way. Recall what a karyotype is. The array of all the chromosomes that you contain as an adult diploid. So remember, these are paired based on the first 22 pairs being our autosomes, and that last pair in the far right-hand lower quadrant, you'll see the sex chromosomes. Note those sex chromosomes. What gender is this individual? You see an X and a Y. That makes this individual a male. Sex determination in humans is that X and Y chromosome, but that's not true for all organisms. Many reptiles, like turtles and crocodiles, it's the temperature at which the offspring are incubated. If eggs are kept too hot, that will drive them all to one gender. So their eggs need to be kept at multiple temperatures to ensure they get both male and female crocodiles for the next generation. But in humans, it's the X and the Y. To be a female, genetically you have to carry two X's. For a male, genetically you have to carry an X and a Y. This XY system is fairly common through mammals, but other organisms also have slightly different sex chromosomes. Birds have known a ZY system, where basically the males are the ones carrying the ZZ and females are ZW. Insects also have a unique, what we call haplodiploidy system, where many of the males only actually are haploids, carrying only one of the sex chromosomes, but also only one copy of every chromosome. Haplodiploidy systems only exist in some insects, such as the group called Hymenoptera, the bees, wasps, ants, and termites. Sometimes we want to look at the genes that are located on the sex chromosomes. In particular, let's look at some X chromosomal genes. So, two conditions I'll ask you to memorize: hemophilia and red-green color blindness. Both of these traits are recessive, and both lie only on the X chromosome, never on the Y. Therefore, when we're solving Punnett squares with genes that are attached to the chromosomes, we'll do a superscripting. In this example, you'll see a dominant big H and a recessive little h. The big H is truly dominant. That means you do not have hemophilia or you do not have color blindness, so normal vision. But the little h is the recessive allele for giving you hemophilia or color blindness. So notice how they've set up this Punnett square. The male at the top has a dominant form of this hemophilia on the X chromosome. But the Y, well, this gene doesn't exist on the Y, so he only has one copy. Look at the female on the left-hand side. She's a heterozygous. See, one of her X's has a big H and one has a little h. This is also known as a carrier. Note that they're children. Let's summarize the offspring. You can tell here not only whether their children have hemophilia or not, but you also get a second piece of information. You get to know what gender. Do they have sons or daughters? So let's start. If we were to summarize the offspring's genotypes, you'll notice that we have X big H, X big H. What's that phenotype? That's a female who does not have hemophilia. We also have another female, big H on an X and little h on an X. This too is a female who is a carrier, but she will not have hemophilia. So both of their daughters are going to appear to be normal or not hemophiliac. But look at their sons. One son has an X big H Y. Does he have hemophilia? No, because he's dominant on the H. But the last son with a little h on the X, well, he's got hemophilia. Hemophilia is known as the bleeder's disease because they have problems with blood clotting. So if we summarize their offspring, they're essentially going to get 2 daughters that are not hemophiliacs, 1 son that is not a hemophiliac, and 1 son that is. They have a 25% chance of having a son who is a hemophiliac. Now, when summarizing, make sure you're accounting for all boxes. And that's why I said 25%, because only 1 in the 4 total boxes is a male with hemophilia.